So let's end the video by talking about the central dogma of biology. So this is where we have information, genetic codes stored in DNA. We're going to make a copy of it, a copy of a gene, an mRNA, and we'll use it to make a protein. Keep in mind, a gene is a segment of DNA used to make one protein. One gene, one protein. The process of moving from DNA to RNA is transcription. So this is where you take your big recipe book of family recipes and a note card, and you're going to write out just the one recipe you need. You're going to transcribe that recipe. It will scribe in a room. Translate is when we're going to switch languages. We're going to switch from the language of nucleotides with our RNA to the language of amino acids in our protein. This is done through translation using codons. So 3 nucleotides will be converted to 1 amino acid, 1 codon. And the transfer RNA carries that amino acid to our ribosome, which is made up of ribosomal RNA. So we've got messenger RNA, transfer RNA, and ribosomal RNA. And so showing you just a brief overview of transcription, we're going to make mRNA in the nucleus of one piece of our DNA template, and then it will leave our nucleus and attach to a ribosome for translation. So transcription occurs in the nucleus, whereas translation occurs at a ribosome in the cytoplasm or on our rough ER. Then that piece of RNA is going to serve as the guide. Transfer RNAs are going to bring in amino acids to our ribosome made of ribosomal RNA, and we'll make a growing chain of proteins corresponding to that chain of messenger RNA. Okay, so we've finished cells. We've finished Module 1. In Module 2, we'll talk about tissues and skin. Please see the PowerPoint if you have specific questions, you want to look at that, and then also see the extra videos I've posted.